Climate change, driven primarily by human activities, is one of the most pressing challenges of our time. Over the past century, the Earth's average temperature has risen at an unprecedented rate. Rising greenhouse gas emissions, such as carbon dioxide and methane, trap heat in the atmosphere, leading to warmer temperatures globally. This warming trend disrupts ecosystems, intensifies extreme weather events like hurricanes and droughts, and threatens biodiversity. Why should we care? Well, apart from the extreme heat we are experiencing this summer alone, this is our home, our children's home, and our children's children's home. As leaders of our planet's future, understanding and addressing climate change is more crucial than ever. At the University of Delaware, we recognize the urgent need to study and mitigate its effects. As mentioned previously, climate change is altering our weather patterns drastically, which leads to unpredictable growing seasons. These changes directly impact crop yields and food security worldwide. As our planet faces the escalating challenges of climate change, one crucial aspect of adaptation lies in our agricultural practices. I worked with Dr. Chi Mu in her lab and field, and we studied the genetics, development, and environmental regulation of agronomical important traits in maize and sorghum. Dr. Mu's goal is to genetically improve crop resilience and production under climate variability. There are many different tasks to do in the field, such as installing soil sensors, as seen here, as well as taking down measurements from those sensors.
40, 34.4, 60, 37.7, 1 meter, 21.3. Correct.
However, our summer did consist of more than just installing sensors and collecting data from them. In order to obtain the main goal of finding our resilient crop, we had to analyze the phenotypes of large quantities of plants made from different genotypes in order to determine resilient traits and where they were coming from. We did this by assessing plant height, stand count, and photosynthesis performance to understand how these crops were adapting to different environments and to understand possible correlations between any of the phenotypes. Plant height gives us a snapshot of growth rates over a certain period of time and under different conditions. Stand count reveals population dynamics and density effects on growth patterns. Photosynthesis performance, measured through chlorophyll fluorescence and CO2 uptake, tells us how efficiently these plants convert sunlight into energy. By analyzing these metrics, we unravel the adaptive strategies of sorghum and maize strains and can identify and select plants with superior resilient traits. Once the genotypes have been identified, the next step involves crossing them to produce a hybrid offspring. This can be done by hand pollination, another task done in the field, and personally my favorite one. Hand pollination is a manual technique where pollen is transferred from the male to the female reproductive organs of the crop by human intervention. The male part of the plant is on top and are called tassels, while the female part is the ear or cob, shown here. The ear grows really elongated stigmas called silks, which are depicted here. Once the ear begins to grow and becomes a good size, you can cut to check to see if silk is begun to form. And if it has, we'll put a white shoot bag in order for the silk to be fresh and ready for the pollination the next day. The silk will grow as shown here, so we don't want the bag to be too loose or too tight. After the ear is bagged, the tassels must be bagged as well in order to collect pollen. It is dated so that we know what day it will be transferred to the ear. On that day, the bag is removed from both the tassels and the ear, and the bag that had collected the pollen is placed on top of the ear and stapled in the back for pollination. In conclusion, hand pollination plays a crucial role in our research here. By manually transferring pollen from the tassels to the silk of the maize and sorghum plants, we ensure controlled breeding and the preservation of desirable traits essential for crop resilience in the face of climate change. This diligent process not only contributes to our understanding of plant genetics and adaptation, but also underscores the importance of innovative agricultural practices in securing food security for our future generations. As we continue our work through the summer, exploring the intricate dynamics of plant growth and adaptation, we remain committed to advancing sustainable solutions that mitigate the effects of climate variability on global food production. Thank you for joining me on this journey through the fields of biotechnology and agricultural science, and also a special thank you for the contributors and supporters of the Envision program. Stay tuned for more videos on ongoing research and discoveries, and remember, the Earth's future is in our hands.